Hi, this is Edward Awad, and welcome to this video about biological diversity part 1. As we have seen in previous videos, scientific evidence has pointed out that life appeared on Earth some 3.6 billion years ago in the form of single-celled organisms that were similar to present-day prokaryotes. The descendants of these ancestral cells have evolved over time and have led to the evolution of a great diversity of life forms on planet Earth, from bacteria and archaea to protists, fungi, plants, and animals. But a discussion on the diversity of life is not complete without first mentioning viruses, which are a special group when compared to living things. So what are viruses? Viruses are not cells. They are rather tiny particles that are dependent on cells for their reproduction. In other words, viruses are obligate intracellular parasites that can produce only within living cells. Compared to cells, viral particles are super tiny. For example, HIV, the virus responsible for AIDS, is 20 times smaller than a typical E. coli cell. When they need to reproduce, viruses use the host cell's biochemical machinery to produce new viral particles. They usually destroy the host cell in this process. The host cell releases progeny viruses, which then can infect new hosts. Outside the cell, the individual viral particles are called virions. Viruses also cannot carry out two basic cellular functions on their own, meaning outside the parasitized cell. And these functions are transport of substances across the plasma membrane and metabolism. It is for these reasons that viruses are not considered as living things and therefore are referred to as agents and are not given scientific names. A virion consists of genetic material, which is either single or double-stranded DNA or RNA, that is generally surrounded by a protein coat known as capsid. Some virions are surrounded by a biological membrane that they steal from host cells as they are released by budding from the surface of their host cells. Virions can take up different shapes, which are primarily determined by the type of capsids they possess. Some virions are rod-shaped, some are polyhedrons with multiple surfaces, others are more spherical in shape, while others possess complex shapes with what looks like heads and tails. There is overwhelming evidence that supports the theories on the evolution of prokaryotes and eukaryotes. This evidence points to three principal ideas. First, that the early living organisms to evolve on Earth were single-celled prokaryotes. Second, that all present-day bacteria and archaea are all unicellular. And third, that the first eukaryotes to evolve were also single-celled organisms. Based on these observations, biologists hypothesize that the early eukaryotic cells had a nucleus, an endomembrane system, and mitochondria, with some cells later evolved to contain chloroplasts. Early eukaryotic cells had a cytoskeleton but lacked a cell wall. The leading hypothesis that explains the origin of the nucleus and the endomembrane system is based on the infolding of the plasma membrane that resulted in the enclosure of chromosomes and the formation of a membrane network as the precursor of the endomembrane system. Of course, this infolding wouldn't be possible without the absence of cell wall and the presence of a flexible cell surface. Now, how did the mitochondria and chloroplasts come to exist? This is explained by a well-known theory in biology known as the endosymbiosis theory. The term endosymbiosis is derived from three Greek word roots. Endo, which means internal, sym, which means together, and bio, which means living. The term symbiosis in biology refers to individuals of two different species that live in physical contact with each other and that are dependent on each other for survival. The theory of endosymbiosis proposes that mitochondria and by extension chloroplasts originated when a bacterial cell took up residence inside a eukaryote about 2 billion years ago. The proposed initial steps involved: first, the eukaryotic cell used its cytoskeletal proteins to internalize a small bacterial cell. The eukaryotic cell could not use oxygen to make ATP and therefore relied on fermentation as a way to oxidize high potential energy organic compounds such as sugars. The bacterial cell, on the other hand, was capable of aerobic respiration, meaning it could use oxygen in order to oxidize high potential energy organic compounds. Compared to fermentation, aerobic oxidation results in a much higher yield of ATP. The engulfed bacterial cell did not fuse with the lysosome and was therefore able to survive inside the cytoplasm of the host eukaryotic cell. The engulfed bacterial cell began to live inside the host cell and to use aerobic respiration to make ATP. The relationship between the host cell and the bacterial cell became stable because of the mutual advantage that existed between them. The host cell provided the bacterial cell with protection and high potential energy organic compounds, and the bacterial cell supplies the host cell with ATP using aerobic respiration. It is believed that chloroplasts evolved in a similar manner through the acquisition of photosynthetic bacterial cells by eukaryotic cells. There are several observations that provide evidence for the theory of endosymbiosis. First, the size of mitochondria and chloroplasts are about the same size as an average bacterial cell. Second, both organelles replicate themselves by the process of fission, just like bacterial cells. Third, both organelles have their own ribosomes and manufacture their own proteins. The ribosomes are very similar to bacterial ribosomes, both in size and composition. Fourth, both organelles have double membranes, consistent with the engulfing mechanism described by the theory of endosymbiosis. And lastly, both mitochondria and chloroplasts have their own genomes, and their genes are very similar to bacterial genes. Mitochondrial gene sequences are much more closely related to the sequences from bacteria belonging to the lineage of proteobacteria than to sequences from the nuclear DNA of eukaryotes. And chloroplast genes resemble cyanobacteria genes more closely than they resemble plant genes. Since mitochondrial and chloroplast genomes became part of eukaryote genomes, the theory of endosymbiosis represents events that led to the transfer and incorporation on a large scale of genes between species. This gene transfer phenomenon is known as lateral gene transfer.